Hi, my name is Eyuche Okorie and I am a NexGeneGirl science intern. My research was at the Yamanaka lab in the Gladstone Institute, and I studied the effects of eukaryotic initiation factor 4G protein families, and I looked at their effects on human-induced pluripotent stem cells. So human-induced pluripotent stem cells, or iPS cells, have two essential properties, which are self-renewal and pluripotency. So self-renewal is the ability to divide indefinitely, and pluripotency is the ability to differentiate into the three different germ layers. So the reason why I was studying these two proteins is because they play a key role in the initiation of translation. And so if you look here, you can see EIF4G1 has this cap binding domain that EIF4G2 does not. But besides that, they're actually pretty similar. And this cap binding domain is responsible, or people believe that it's responsible for translation initiation. And so what I did was compare the two functions between these proteins to see how specifically they interact and play a role in translation. So the first strategy I used is a conditional knockdown, where I introduce a guide RNA vector into our Cas9 locus, and what we saw was that upon addition of DOX, the expression of these proteins decreased over time. And when we look at the phenotypic effects on self-renewal, we actually see that both of these proteins are proliferating less, but EIF4G2 is affected more drastically, where we're not seeing much proliferation at all. And then when we look at the morphology, we see a drastic change in the cell shape. They're smaller and a lot more deformed. And then when I looked at the phenotypic effects on pluripotency, what I saw is that when we didn't add DOX, EIF4G1 had its PAX6 marker affected, and PAX6 is the marker for ectoderm, so we think that EIF4G1 is responsible for differentiation of neurons and different neural cell types. And because we were afraid of residual protein expression or escaper cells, we did a second strategy, which is our conditional knockdown strategy, where we transfected in a DOX-inducible transgene, and we use neo and puro selection targeting in order to make our conditional knockout cells. And then when we look at our wild type, with and without DOX, we actually see that there's still expression of EIF4G2. But when we look at our endogenous, or our knockout cells, we're seeing that there's only expression of 4G2 when we add DOX, but when DOX is not present, there's no expression, which means that we actually can validate our results since we have our DOX-inducible transgene, and there's only one reason why we wouldn't see any expression. In the future, we also aim to identify other unknown pathways that these proteins play a role in, and hopefully these efforts will help us in elucidating the mechanisms behind essential stem cell properties. I would like to acknowledge Songnan Wang and Shinya Yamanaka for this amazing opportunity, and I would like to give a special thanks to the NexGeneGirls program for allowing me to have done this spectacular research.